In this lecture, we are going to start going through biochemistry. If you remember, biochemistry is the study of carbon-based compounds that are produced by biological entities. So that covers proteins, well, the ones we're going to look at anyway, are proteins, lipids, and carbohydrates. So first, we're going to look at proteins. Now, all proteins have some basic characteristics. They all contain at least carbon, hydrogen, oxygen, nitrogen. They're all based off of amino acids, and that's the basic components of amino acids. Now, what makes proteins different and amino acids different are the radical groups that come off of them. So there's side chains on all amino acids that kind of give them specific properties and characteristics. Proteins are very large molecular mass. They're very, very, very big polymers of amino acids, as I just said, polymers of amino acids. They all will exhibit a characteristic called imbibition. Imbibition is the ability of proteins to suck in moisture from their surroundings. That will make the proteins swell, and the proteins will oftentimes soften if it stays that way for too long. Again, this imbibition is the proteins absorbing moisture from their surroundings. This is what happens when you sit in the tub for too long and you get all pruny. That's imbibition. That's your proteins sucking in moisture. Now, again, there is some biological need for imbibition, but imbibition that goes on for too long leads to, especially in like a dead body, is going to lead to very rapid decomposition. Also with proteins, if we treat them with chemicals or through heat, they'll go through coagulation. That's conversion of soluble protein to insoluble protein through heat or chemical means. Now, we tend to use aldehydes. You could also use alcohols or phenol. Another term for coagulation is cross-linking, at least when we're using aldehydes. But through heat too, again, that's what happens when you crack an egg into a hot pan. It solidifies, it's the proteins coagulating. This is also why if you watch a body get cremated, their arms and legs will start to move because you're heating up the proteins, they're coagulating, and they're going to contract. So if you look at the function of proteins, at least in the human body, they are the structural components, makes up about 16% of body mass. So again, this is one of the things that why we say that embalming is temporary. The way we embalm, we're only embalming at best 16% of the body. The rest is carbohydrates, lipids, bone, and our embalming does not touch any of those things. So that's fairly important to us. But also enzymatic action. So enzymes are proteins. And if you remember back to decomposition, you have hydrolytic or autolytic decomposition that has to have enzymes for it to work. So if I can deactivate the enzymes, I can slow down decomposition. And I can do that because the way we embalm coagulates the proteins. So enzymes get kind of shut down. Not so much important to us is motility. Motility is the ability of microorganisms to move. So any mobile microorganisms, their motility functions, their flagella are protein, and that is one way that we can help to disinfect a body is that if I can shut down the motility of microorganisms, they won't spread as easily. Proteins stabilize and control activity of nucleic acids. That's not so much anything that we concern ourselves with. That's more the functioning of cells when they code for proteins. They're making nucleic acids. That's your DNA, RNA, that kind of things. And so proteins, your cells are little protein factories, and so they will help to stabilize and control that activity. They're important in cell membrane transport and recognition. Again, not something that we're necessarily concerned with. That's more of a physiological living function. They do have some extracellular importance in that proteins make up collagen. Collagen is the main protein of animal connective tissue. So collagen is kind of what holds you together. It's a cementing mechanism. So proteins are important for keeping you together. Again, also not terribly important for us are hormonal. Most hormones are proteins. But what could be important to us are toxins. There are bacteria that make toxins that can make embalming a body a little bit more dangerous. Again, if I can make those toxins inactive by using formaldehyde or glutaraldehyde, then that's going to make the body a little bit safer and it's disinfecting it. So, as we stated, proteins are polymers of amino acids. So proteins are just this made up of this unbranched amino acid chain. Most commonly, most proteins are going to range between 50 and 1,000 amino acids. Some as many, some of the biggest proteins are up to 50,000 amino acids. There's also proteins that are as few as 3, but most commonly you're going to find between 50 and 1,000 amino acids in this chain. Proteins get initially kind of made up of 20 different amino acids. They get put together in different combinations to make a specific type of protein. And once they get assembled, they can be modified after that too. So you have about 20 different amino acids that put in different sequences make up the vast majority of proteins. So looking at the structure of an amino acid, that's the basic structure of all amino acids. They all have, again, hydrogen, nitrogen, oxygen, carbon. And so the only difference between them is that little R group right there. So you have, and why they call them amino acids is moving left to right, you have the amino group, which is, again, the ammonia group. That's the group that formaldehyde and glutaraldehyde will react with. You have the alpha carbon, which is how it kind of rotates, and you'll see that after a bit. The alpha carbon is also where the radical group is attached, and the radical group is what gives an amino acid its identity. Then at the far end, you get the carboxyl group. Again, that's the carboxylic acid. That's why they're called amino acids. And all of these are pretty much in straight chains, except so they all pretty much have this basic amino alpha carbon carboxyl end, except there's one called proline. Proline is an oddball in that it has a ring configuration, but it's the only amino acid that does. Now looking more closely at the radical group, as we said, this is what gives the amino acid its distinct identity, its distinct chemical properties. It can be any number of things. If again it's hydrogen, it's glycine. As mentioned before, glycine is the simplest of amino acids. It can have more groups than just that though. So it can have more complex molecules containing things like oxygen, nitrogen, or sulfur. Again, the sulfhydryl groups are what is where you'll get thiols or mercaptans from decomposition. So it can give that a dead body kind of a rotten egg smell. That's where it would come from, is this radical group. They all have carbon chains on them also. But again, what comes off that carbon chain could be oxygen, nitrogen, sulfur, just kind of depends. So as I start to take these amino acids and I start to link them together to make this chain, this protein, I'm going to create what's called a peptide linkage. A peptide linkage is a covalent link that you create between the amino group of one amino acid and the carboxyl group of another. And what will result is water. So you're going to take the hydroxyl off off the carboxyl end, and you're going to take a hydrogen off the amino end, and they'll link up, and then you end up with a water molecule. And that's why this is considered what's called a condensation reaction or dehydration reaction. As the amino acids come together, water comes out. Any other amino acids that get stuck to this chain are always going to get added to the carboxyl end. That's always where they go. They never get hooked to the amino end. For whatever reason, it has to do with charge polarity, but it's always going to get attached to the carboxyl end. Now, amino acids and proteins for that matter, but amino acids specifically, are amphoteric. If you remember, amphoteric means it can act as either an acid or a base. And this is because on one end you have a carboxyl group, which is an acid, and on the other end you have an amino group, which is basic. So again, that's the carboxyl end. If you put that in solution, that's going to create hydronium. The hydroxyl group comes off, or I'm sorry, the hydrogen of the hydroxyl group comes off, attaches to the unpaired electrons in the oxygen molecule, and you end up with H3O plus hydronium. That's one of the definitions of an Arrhenius acid. With the amino group, you put that into solution. It's going to take a hydrogen proton off of water, and you end up with ammonium technically, but the NH3 plus, and then the water molecules can be left with an OH negative. Increase of hydroxyl ions is a definition of an Arrhenius base. So because they're amphoteric, amino acids will act as buffers in solutions. So they'll be able to neutralize both acids and bases if they're in a solution. And again, because proteins are polymers of amino acids, they can do that also. So we looked at the structure, basic structure of an amino acid. Now we're going to look at the basic structure of a protein. Proteins have 4 basic structures. The very first one, the most simple, is the primary structure, which is just a sequence of amino acids. It's different for every protein because, again, you take about 20 different amino acids, but then you stick them in different sequences, and that's how you get a particular type of protein. So just linking together a bunch of amino acids is the primary structure of the protein. But when you do that, when you link all these amino acids together, they start to twist around, and that's called the secondary structure. So the twisting of the polypeptide. Polypeptide means, peptide indicates amino acids. Polypeptide means a lot of amino acids. And so once you get the primary structure together, it starts to twist, and it always twists around the alpha carbon. And the reason it's always going around the alpha carbon is because it takes way too much energy for it to try to twist around the peptide linkage. And so as it rotates, this rotation is actually caused by hydrogen bonds. If you remember, hydrogen bonds are bonds between hydrogen in one molecule and unpaired electrons in another molecule. And so this is a hydrogen bond that forms between the amino group hydrogens, so the hydrogens on the amino groups, and the carboxyl groups of oxygens. Sorry, the oxygen of carboxyl groups, that's a better way to put it. And that's where it's going to start to twist, and it makes this helix. Okay? And the number and position of these bonds kind of helps to determine what the secondary structure is going to look like, because technically there are 2 types of secondary structures. Okay? And so again, however many hydrogen bonds there are and kind of where they are will determine the secondary structure. And you can have either an alpha helix, which is kind of what many people think of as protein structures. This is what the structure of DNA does. It's a double helix, but this is just a single. So that's the alpha helix, or what's called a pleated sheet. But again, it's twisting around that alpha carbon, not around the peptide linkages. So once I get the secondary structures created, then I can start to stick them together to make what's called a tertiary structure. Tertiary structure of a protein is the attraction between different secondary structures. And this is what gives the protein its function. So how you put these different secondary structures together to make this tertiary structure will determine what the protein can do. This also helps to create what's called a hydrophobic core. So the hydrophobic core, hydrophobic means doesn't like water, is one of the things that we'll talk about a little bit later when we talk about protein fixation. The hydrophobic core can be disrupted by some types of preservatives, and that which helps to preserve the protein. So once I've put all these secondary structures together, made the tertiary structure, I can then take tertiary structures and make quaternary structures. Quaternary structures are proteins consisting of 2 or more amino acid chains. Technically, it's 2 or more tertiary structures. Now, why this is kind of important is that preservatives, mainly formaldehyde that we use, don't necessarily fixate all of these different types of protein structures. It may do some, but it won't necessarily do others. And so again, that's why we talk about temporary preservation. No matter what we do, We can never get all of these different protein structures to fixate, and that can cause us problems in the future. Okay, so that finishes up what we're going to talk about this time. Next video is we're going to talk about different types of proteins and enzymes and their actions.